Hi, everybody. Good day, good morning, and good evening for those of you who are in a different time zones than us. Thank you very much for coming to our ZOOMinar series today. We will have two speakers, Dr. Eri Chatani and Dr. James Shorter. Each talk, as usual, will be 30 minutes, followed by about 10 minutes questions and answers. And at the end of the second Q&A session, we will be letting people into the panel if they want to ask their questions in person. So we will have a continued discussion on the speakers' talks. It is my great pleasure to introduce Dr. Eri Chatani. She is an associate professor at the Department of Chemistry, Kobe University. Dr. Chatani did her PhD in the lab of Professor Rikemaru Hayashi in Kyoto University. And during her PhD, she studied the properties of native proteins using site-directed mutagenesis and X-ray crystallography. She did her postdoctoral studies in the lab of Professor Yuji Goto, where she was introduced to the amyloid research, which she continued doing in her own independent later on studies. I don't want to take much time from you. It was really nice meeting you, and glad that you can participate in our ZOOMinar series. It's your turn now.
Thank you very much for your kind introduction. So, can you hear my voice?
Yes.
Yes, you're fine.
Thank you very much. So, let me begin my talk. Hello, good afternoon. I am Eri Chatani from Kobe University. First of all, I thank Professor Ramamuzi and the organizing members for the kind invitation. I am very happy to have a great opportunity to talk in the ZOOMinar. I have been working on physicochemical properties of amyloid fibrils and their formation. Recently, I am interested in amyloid nucleation. So today, I'd like to talk about our investigation of protein assembly in the nucleation of amyloid fibrils. Amyloid fibrils are a kind of aggregate made of proteins or peptides. You know, their structure is typically highly ordered. They show needle-like morphology, hierarchical structure composed of protofilaments, and inside them, cross-beta structure can be seen. And recently, solid-state NMR and cryo-EM have determined a lot of beautiful atomic structures of many amyloid fibrils. These are several examples taken from literature. As shown here, polypeptide chains are folded into a planar structure to make layers and then stack along fibril axis. Based on these structures, we can say that amyloid fibrils are a kind of supramolecular assembly with crystal-like ordered structure. Due to the highly organized and periodic structure of amyloid fibrils, there are similarities between crystals and amyloids in formation mechanism. This illustrates a basic scheme of amyloid formation. It is composed of nucleation and elongation phases. In the nucleation phase, amyloid nuclei are supposed to be formed. After that, the reaction goes into elongation and the amount of amyloid fibrils grow rapidly by attaching monomers at the end of the nuclei. And as seen in general crystallization, amyloid nucleation functions as a rate-limiting step and plays a very important role in the initiation of the reaction. So, in this study, in order to understand the detailed mechanism of amyloid nucleation, we raise a question of how protein assembly proceeds during nucleation. If we can observe the processes of protein assembly to nuclei like this cartoon, they will become much help to understand detailed processes. However, according to a classical nucleation theory proposed in crystallography, nucleation is considered to be a reaction that passes through a single energy barrier, and so it is theoretically predicted it is difficult to observe any stable intermediates. Here, let me explain about the classical nucleation theory briefly. To my knowledge, this is the simplest theory used to study energetic properties of crystalline nucleation. In this theory, the energy for crystallization is described by the sum of interfacial free energy and bulk free energy. For example, when the crystal shape is assumed to be a sphere, the free energy can be described like this equation. And this orange curve is the plot of this equation. The interfacial free energy has positive values, and the bulk free energy has negative values, and therefore they cancel each other as a function of the crystal radius. When the crystal is small, the interfacial energy is dominant, and the total energy shows a positive value. But as the size grows, the bulk energy becomes dominant, and the energy finally decreases. An important feature of this crystallization energy is that there is no local minimum during crystal growth. So, based on this theory, it will be difficult to capture some dynamically stable intermediates during nucleation. And this theory has also been applied to chain polymers forming anisotropic crystals. The basic idea is exactly the same. Here shows an example of a polymer crystal in which a polymer chain is folded into an anisotropic shape. In this case also, when the total free energy is described as the sum of interfacial free energy and bulk free energy, no local minimum never appears within the energy diagram. Nevertheless, there are many experimental reports suggesting the formation of oligomers and protofibers before nucleation. Here are some examples taken from literature. There are oligomers, protofibers, and furthermore, annular oligomers. These species have been reported both in vitro and in vivo for many proteins. When looking at these aggregates, it is natural to predict that some of them may act as nucleation intermediates. However, this idea is inconsistent with the classical nucleation theory I mentioned before. This is quite controversial, and we have to say that exact mechanisms of nucleation is not fully understood. With this situation, we are motivated to see the details of amyloid nucleation, such as what kind of aggregation proceeds during nucleation, and whether early aggregates contribute to nucleation or not. Today, I'd like to talk about our observations using insulin or insulin peptide as models. I have two subjects. The first one is about the formation of protofibril-like aggregates that plays a role as a nucleation intermediate. And I will also talk about the effect of amino acid sequence on early aggregation and the mechanism of protofibril formation as a second topic. Let me start with the first topic. In this study, I investigated the amyloid formation of insulin V chain. The V chain is a part of insulin, and it is a peptide made of 30 amino acid residues. This peptide was already reported to form amyloid fibrils by Professor Fink's group. And actually, with the help of shaking, we could see the amyloid formation very clearly with typical sigmoidal shape of thioflavin T intensity. Interestingly, in the lag time, a large amount of curvy aggregates were found. They looked like protofibrils, and they gradually changed to amyloid fibrils with time. To better understand the reaction scheme in more detail, we proceeded investigation. In the fibrillation in the previous slide, we used shaking to enhance the reaction. When the same sample was incubated without shaking, the increase in thioflavin T fluorescence became very low. And from the analysis of electron microscope, it turned out that under quiescent conditions, protofibrils accumulated without changing into amyloid fibrils. This result suggests that the protofibrils are metastable and require agitation to change into amyloid fibrils. Thanks to the metastability of protofibrils, we could track their formation process nicely by using far UV CD spectra and DLS. And these are summaries of the time-dependent changes of CD and DLS measurements. They both showed by exponential patterns, and from them, it was suggested that the protofibrils evolved their size and structure in a stepwise manner. We also conducted a time-lapse SAX measurement to know more about the way of evolution. The overall shape of the SAX profile supported the rod-like shape of protofibrils. By analyzing these profiles together with the DLS data, the time-dependent changes in diameter and length of the protofibrils could be estimated. And this is a summary of our observation, suggesting the protofibril size and structure evolved in two steps. And after the protofibrils are formed, they change to amyloid fibrils by applying agitation. Interestingly, the structural transformation is triggered not only by continuous shaking, but also a short-term ultrasonication. When a single short ultrasonic pulse was given to the protofibrils, their secondary structure began to change dramatically to needle-like amyloid fibrils. It was amazing to see the transformation was triggered just with one short ultrasonic pulse. Here, when the sonication-triggered fibrillation was investigated in more detail, we found a difference in fibrillation abilities between the first and second protofibrils. From a series of investigations, we could see the rate of fibril growth changes depending on the state of protofibrils. This is a summary of the growth rate, and what we observed here is that no fibrillation occurs when the first protofibrils are dominant, but fibrillation starts after the second protofibrils are formed. It means that the second protofibrils, not the first ones, play an important role as a nucleation intermediate. This is a schematic illustration of amyloid formation of the B chain proposed in this work. What we'd like to suggest here is that the nucleation of the B chain amyloid fibrils is multi-step. First, the B chain formed protofibrils in 2 steps. The second protofibrils can function as a nucleation intermediate. However, there is a large energy barrier. To overcome it, it is necessary to supply energy from outside, such as shaking or ultrasonic pulse. Once the energy barrier is crossed, nucleation and subsequent amyloid formation is supposed to occur. To compare with the classical one-step nucleation, we tried to make another type of energy diagram. I know this is oversimplified, but I think this may be useful to get what happens in multi-step nucleation. You can see there are 3 different energy curves. The green is for the first protofibrils, and the blue is for the second protofibrils, and the red is for amyloid fibrils. As indicated by solid arrows, we may be able to understand that protein aggregation proceeds in a multi-step way by crossing several different energy curves toward more stable state and finally reaching amyloid fibrils. Okay, I'd like to move on to another example of insulin. In this experiment, we found that there is a strong sequence dependence on the formation of protofibrils. We also tried to investigate the mechanism of protofibril formation. In this study, we used full-length insulin from 2 different sources, bovine and human. Their amino acid sequences are basically similar, but there are 3 different residues out of 51 amino acid residues. We previously found that bovine insulin forms protofibril-like aggregates before nucleation at high salt concentration. In this case, the shape of cyclovin T-time course becomes very characteristic like these plots, and the high fluorescence intensity indicates the accumulation of protofibrils. The protofibrils showed metastability, and in the present case, high temperature enhanced the transformation to amyloid fibrils. On the other hand, when we tested human insulin, we found it showed a typical sigmoidal pattern with a lag phase. We initially thought there is no significant difference between them, but unexpectedly, aggregation behavior seemed quite different. We further tracked time-dependent changes in secondary structure using FTIR. In this experiment also, difference was observed between bovine and human. In bovine insulin, a beta sheet peak appeared shortly after the reaction started, and then another peak developed thereafter. In contrast to this, in human insulin, no change was found in the lag phase, supporting the absence of protofibril formation. We further tracked change in size by DLS measurements. Here are results which are shown as size distribution. In bovine insulin, you can see a clear sliding of a single peak with time. On the other hand, there was no change in the main component in human insulin. This observation shows that only bovine insulin started early aggregation. You may think aggregation may also occur in human insulin by looking at this peak. But this graph is an intensity plot, and so its volume fraction was estimated quite small, less than 0.1%. So we ignored it in this work. And to further make sure that the formation of protofibrils was specifically seen in bovine insulin, we checked amyloid formation in a wider range of salt and protein concentrations. Here are phase diagrams obtained from such investigations. In both insulins, amyloid fibrils were formed in a wide range except for a green area where amorphous aggregates were formed. The magenta area is the conditions forming the protofibrils, and the cyan area is the condition where fibrillation occurred without forming protofibrils. As you can see here, the magenta area was not seen in human insulin, and it became clearer that the formation of protofibrils was specifically seen in bovine insulin. This is a summary of the difference between bovine and human insulin. The aggregation process of bovine insulin was multi-step, and we were able to see the formation of protofibrils in the lag phase as seen in the B chain. On the other hand, the aggregation process of human insulin was one step. Early aggregation never proceeded. It is interesting to see that even the slight difference in amino acid sequence determines the presence or absence of early aggregation. Here, as a last subject, I'd like to return to the DLS result obtained for bovine insulin again. The data nicely showed gradual size evolution, and furthermore, each peak showed monomodality. Thanks to such properties of the early aggregation, we could obtain time-dependent change in the association number and diffusion coefficient by the processing of the light scattering data. Both of them showed a characteristic time dependency, and we tried to establish an aggregation model that explains both of these data. To establish an aggregation model, we focused on Smolkovsky aggregation kinetics, which is one of the basic models used for colloid assembly. This model assumes aggregation between two particles. Particles of any pairs of size can aggregate, and aggregation rate is described by this differential equation. Based on this basic model, we tried to reproduce the experimental data. I'm not going into details of the procedure today, but we divided the whole reaction into two stages by looking at the fractal dimension as an indicator to identify the state of packing or morphology of aggregates formed. And we hypothesized that the first stage is the growth of spherical aggregates, and the second stage is their extension to rod-like aggregates. This is a schematic illustration of each stages. The difference between the first and second one is the way of the two-particle association. In the first stage, two particles associate from any directions to form isotropic aggregates. In the second stage, only both ends of each particle are allowed to associate to form anisotropic shape. We switched from the first stage to second stage when the aggregate became a defined size. Using the modified Smolkovsky kinetic model, we attempted to reproduce the experimental data. This is still an ongoing project and needs optimization, but at the moment, we are successfully reproducing our data. This is the assembly process proposed by modeling. The basic scheme is colloid-like aggregation, and insulin monomers seem to first aggregate into spherical particles, and afterwards they attach end to end to form rod-shaped protofibers. This is a summary of our study. From the analysis using V-chain and insulin protein, multi-step pathways of amyloid nucleation through protofiber-like aggregates have been proposed. From our observations, it was suggested that amyloid nucleation is not always one step, but multi-step pathways can also be seen depending on proteins and reaction conditions. This cartoon represents nucleation pathways of the one-step and multi-step nucleation processes represented in the plane of aggregation size and structure ordering. In the case of one-step nucleation, aggregation and structure ordering happen simultaneously, so the path follows the diagonal lines of this graph. On the other hand, when aggregation occurs first and structure ordering occurs afterwards, the pathway will become multi-step through protofibrils. Whether the nucleation takes the conventional one-step or the multi-step scheme may depend on reaction conditions. And furthermore, the result of insulin suggested that amino acid sequence is another important factor to determine the pathway. Our other results further suggest that the number of steps also changes sensitively depending on reaction environments. Interestingly, a two-step nucleation hypothesis has been proposed recently in the field of crystallography, and multi-step nucleation mechanisms attract much attention. This is a schematic cartoon representing the classical nucleation model here and the new two-step nucleation model. In the classical nucleation, solutes are arranged in one step to form a crystal structure. This is what I introduced first as the simplest mechanism of nucleation. On the other hand, in the two-step nucleation, solutes form clusters and crystalline structure appear from the clusters. This scheme has been experimentally proven in colloidal model system and protein crystallization and so on. When we looked at the two-step nucleation in crystallization, it may be very natural to think that multi-step nucleation is another important mechanism in addition to the conventional one-step one. The clusters of solutes and the protofibrils observed in our study may have commonalities, or there may be different features. So we are planning to investigate the structural features and formation mechanism of nucleation intermediates with a wider range of proteins as future research. This is my last slide. This is a page for my acknowledgement. I am supported by many collaborators as listed here. Especially, I'd like to thank the lab members at Kobe University who progressed experiments of B-chain and insulin. I'd also like to thank Hiroshi Imamura, who gave us much help when establishing the aggregation model. And Professor Inoue and Sugiyama supported much when doing SAX measurements. And I also thank all of people for a lot of support. Thank you very much for kind attention.
Thank you very much for the nice talk. I see that we have questions already coming, and the first one is from Ashutosh Tiwari, and it says, a very nice talk relates to B-chain aggregation experiments. Did you modify the cysteine residues on the B-chain of insulin, or it had free SH groups? If not, did you see any disulfide cross-linked aggregates or multimers?
Yes, as you told, B chain has two free cysteine residues, and we did no modification with them. So the cysteine is present as with their free chiral groups, and we confirmed that no SS bonding occurs during the reaction by comparing the reaction in the presence or in the absence of reductant. So no effect of disulfide bonding were involved in our case.
And you have another question which Rams posted in a chat. He says, excellent talk. It is correct that the main difference between first and second protofibers of B chain peptides is the secondary structure. Can the second protofibrils convert or cross-seed the first protofibrils to form fibers without application of ultrasonic pulse?
Yes. Okay, thank you for the question. As for the structural difference between the first and second protofibers, we just know that the second protofibers contain more amount of secondary structure compared to the first one. So, yeah, we need more detailed information, but at the moment, that's all. And as for the second question, yes, when we incubated the second protofilament, when we incubated the second protofibrils under quiescent conditions for a long time, it finally converted to amyloid fibers. But it took very, very long time, almost a month. But anyway, it finally converted to amyloid fibers without ultrasonic pulse.
Thank you very much. We have one last questions before we move to the second speaker. It is posted by Aphrodite, and she says, thank you for the very nice presentation. Is the double nucleation mechanism of amyloid formation similar to the earlier proposed nucleation conformational conversion NCC mechanism, which predicts the oligomers of stable assembly in intermediates?
Yes, thank you for this suggestion. So, exactly, our proposed mechanism is quite similar to the nucleation conformational conversion mechanism. Yeah, that's true. So initially, some oligomerization or some association occurs, and by using this species, nucleation occurs in a stepwise manner. So there is a very commonality between the NCC mechanism and our multi-step mechanism.
Thank you very much. If you can please stay like after the second talk for the
Okay.
for the
Thank you very
discussion.
much.
And we are ready to move to our second speaker, Dr. Jim Shorter. Dr. Jim Shorter is a professor of biochemistry and biophysics and Parallel School of Medicine at the University of Pennsylvania. Dr. Shorter graduated with distinction from University of Oxford, and he then joined Graham Warren's laboratory, where he studied the molecular mechanisms of the Golgi architecture in inheritance, and received his PhD in cell biology from the University of London. Then he moved to the United States to continue his studies with Professor Graham Warren as a postdoctoral fellow in the cell biology department of Yale University School of Medicine. 2 years later, he joined Susan Lindquist's laboratory at the Whitehead Institute for Biomedical Research at MIT. Dr. Shorter is a recipient of many awards, among which are NIH Director's New Innovator Award, Ellison Medical Foundation New Scholar in Aging Award, Grand Challenges Explorations Award from the Gates Foundations, and many others. He has been extremely prolific, authoring more than 173 publications. And without a further delay, it's welcome to our series, and I'm so glad that you can make it and give a talk.
Great. Yeah, so thanks so much for the invitation and a very nice introduction. And so, yes, I'm excited today to tell you about some of our latest thoughts on how to counter deleterious phase transitions in ALS and FTD, and I'll explain exactly what I mean by that. So the 2 neurodegenerative disorders I want to talk about today, amyotrophic lateral sclerosis, or ALS, and frontal temporal dementia, or FTD, are 2 related neurodegenerative diseases which actually sit at opposite ends of the clinical spectrum of disorders. And the opposite ends are actually very different diseases. So ALS, of course, presents with motor neuron degeneration, where both upper and lower motor neurons will degenerate. This leads to initially weakness and spasticity, followed by muscular atrophy and ultimately paralysis, and typically death within 2 to 5 years of diagnosis. At the other extreme of this spectrum sits FTD, where a very different set of neurons degenerate. Cortical neurons in the frontal and temporal lobes degenerate in FTD, which gives rise to a very severe dementia, and patients will typically die about 5 to 10 years after diagnosis with FTD. But what we've realized over the past 10 or 15 years or so is that patients will sometimes present with both phenotypes of ALS and FTD, whereas other patients will be more on the ALS side of things, but with some signs of dementia, whereas other patients still present mostly with dementia, but will have some signs of motor neuron degeneration. Now, the reason for this connection has become clearer as we've unraveled the pathology and genetics that underlie these disorders. And so for example, mutations in the C9orf72 gene, specifically a G4C2 repeat expansion, will give rise to ALS and FTD about 50% of the time, and often patients will present with both phenotypes. At the other extremes, for example, tau, mutations in tau will only ever give rise to FTD, whereas mutations in SOD1 at this end of the spectrum will only ever cause ALS. But the 2 genes and proteins that I'll focus on today, FUS and TARDDP or TDP43, are both RNA binding proteins with prion-like domains that mutations in can give rise to ALS and more rarely FTD. So not only are the diseases connected by genetics, they're also connected by pathology. TDP43 and FUS are typically nuclear RNA binding proteins that reside in the nucleus. But in the context of degenerating neurons of ALS and FTD patients, what we see is that TDP43 has now mislocalized to the cytoplasm, where it's forming these aggregated structures in degenerating neurons of ALS and FTD patients. Now, this phenotype is very common in ALS. About 97% of ALS cases present with TDP43 proteinopathy. And typically in ALS, it's the wild type version of TDP43 that's getting into trouble, and mutations in TDP43 that cause ALS are comparatively rare. In FTD, we see this type of TDP43 proteinopathy in about 45% of cases. Now, FUS can experience a similar type of change where it's ordinarily found in the nucleus, but in the context of these degenerating neurons, accumulates in these cytoplasmic inclusions. Now, this is a rare phenotype in ALS. About 1% of ALS patients present with this type of FUS proteinopathy. And typically in this case, it's a mutant form of FUS that's getting into trouble. By contrast, this type of phenotype is a little more common in FTD. About 9% of FTD cases present with FUS proteinopathy. And here in FTD, it's typically the wild type version of FUS that's getting into trouble. So let me tell you a little bit more about these two RNA binding proteins. So they're both nuclear RNA binding proteins that engage RNAs through RNA recognition motifs. TDP43 has 2 of these. FUS has 1 RNA recognition motif. But FUS also engages RNAs through these RGG boxes in the zinc finger domain. Now, typically they reside in the nucleus, but they are constantly shuttling in and out of the nucleus. They both possess a nuclear localization sequence such that they will be transported back to the nucleus if they go out into the cytoplasm. Now, together they function in pre-mRNA splicing, transcription, RNA transport, and RNA processing, and are really regulating the expression of thousands of human genes. And so there's a definite thought in the field that the loss of the nuclear localization of these two proteins leads to dysregulation of thousands of human genes, and this contributes to the neurodegeneration. In disease, they're mislocalized to these cytoplasmic aggregated structures in degenerating neurons, and these aggregated structures are also likely conferring a toxic gain of function. So in both ALS and FTD, we're really fighting against a loss of nuclear function of these two RNA binding proteins, but also a cytoplasmic gain of toxic function. Now, mutations in TDP43 or FUS can cause ALS and FTD, but the initial thing that really leapt out to us about these two RNA binding proteins was the presence of what we call a prion-like domain at the C-terminal end of TDP43 and the N-terminal end of FUS. Now, these are very distinctive low complexity domains that typically only contain a few amino acids, which are typically uncharged polar amino acids like glutamine, asparagine, serine, and tyrosine. And then aside from those residues, you really don't find many other amino acids in these types of domains apart from glycine. Now, this domain gets its name because of its similarity in amino acid composition to prion domains that enable various yeast proteins like SUP35 to form prions in yeast. And in the context of SUP35, all the information needed to form a prion is encoded in its prion domain. And so although we don't know whether TDP43 and FUS really form prions in the same way as SUP35 and other yeast prion proteins, they can certainly form self-templating fibrils in the test tube, and these can elicit disease phenotypes when they're injected into the brains of transgenic mice, for example. And it's really the prion-like domain that underlies this ability to form self-templating fibrils. So if we delete the prion-like domain of TDP43 or FUS, then they'll lose the ability to form these fibrillar structures. Now, at the same time, over the last 5-10 years or so, what we've realized that prion-like domains also enable the proteins to do is to undergo this process of liquid-liquid phase separation, where TDP43 or FUS will switch from being monodispersed in solution into a condensed liquid phase that we believe underpins the formation of various functional membranous organelles, particularly inside the nucleus of cells. And so the idea goes that this is a functional structure, and by creating a specialized microenvironment in this liquid phase, TDP-43 and FUS are more able to catalyze their or promote their biological functions. And so, for example, if we delete the prion-like domain of TDP-43 such that it loses its ability to undergo liquid-liquid phase separation, TDP-43 will also lose functionality in about 20 to 30% of its pre-mRNA splicing reactions when surveyed in a genome-wide manner. Likewise, FUS will lose functionality if it's unable to phase separate in processes like DNA repair. And so this, we believe, is really the beneficial function of the prion-like domain in TDP-43 and FUS. But this process places prion-like domains in close apposition in these liquid phases that puts them at risk of forming more stable structures. And eventually, over time, these liquid droplets will convert into gel-like structures and ultimately into the pathological fibrils that we associate with the end stages of disease. And here, the fibrils present with a cross-badal-like architecture. And so it's really this final step of this phase separation reaction scheme, this liquid to solid phase transition that tends to be accelerated by disease-linked mutations in TDP-43 and FUS. And so I want to stress that this doesn't necessarily imply that all liquid to solid phase transitions are deleterious. For example, we know that TDP-43 seems to form a functional amyloid in muscle cells during muscle regeneration and development. TDP-43 seems to form a functional amyloid-like structure. But in the context of neurodegenerative disease and ALS and FTD, we really have a lot of evidence now that this transition to the solid phase is a damaging process that underlies this neurodegeneration. And so what the lab is very interested in doing is uncovering therapeutic protein disaggregases that can reverse this reaction scheme and recover monodispersed TDP-43 or FUS in solution and restore them back to the nucleus where they can perform their regular function. And so in essence, reversing the formation of these pathological aggregates, these solid phases, but also the liquid phases to allow us to reset this phase diagram and restore TDP-43 and FUS homeostasis. And today I'm going to focus on one class of disaggregases that we've uncovered in recent years, the nuclear import receptors for FUS and TDP-43 that really have a very potent ability to drive this reaction in reverse and convert fibrillar forms of TDP-43 and FUS back to soluble TDP-43 and FUS. So we're going to focus mostly on carioferrin beta-2, also known as transportin 1. I'll call it Cat beta-2. And the Cat beta-2 is the nuclear import receptor for FUS, as well as several other prominent RNA binding proteins with prion-like domains that are connected to neurodegenerative disease, such as TAF15, EWS1, HNRMPA1, and HNRMPA2. What Cat beta-2 does is it comes and engages the PY-NLS sequence of FUS. So FUS has a distinctive nuclear localization sequence that we call a PY-NLS because of this proline tyrosine motif at its C-terminal end. And what Cat beta-2 does is come and engages this NLS sequence with very high affinity, with nanomolar affinity. Cat beta-2 is a superhelical protein comprised of 20 heat repeats that really cradles the FUS PY-NLS in this region here, making multiple contacts with the FUS PY-NLS. And this is a very tight nanomolar interaction. And it's the very tight nature of that interaction that initially made us wonder whether Cat beta-2 might chaperone and even disaggregate these proteins, because one thing protein disaggregators have to do when they're pulling proteins out of aggregates is hold onto them very tightly, at least transiently. But then the other thing that made us wonder whether carioferrin beta-2 might serve as a disaggregase for these proteins was related to its ability to function in nuclear transport. So nuclear import receptors are tasked with delivering cargo across the nuclear pore complex and into the nucleus. And this, it turns out, is a non-trivial task, because the interior of the nuclear pore complex is another type of condensate comprised of FG-rich nucleoporin proteins that generate a viscous liquid or hydrogel structure inside the nuclear pore complex. And it's this phase-separated state that actually serves as a selectivity filter for entrance to the nucleus from the cytoplasm. So if you're a protein over a certain size, you will not be able to penetrate through this gel-like structure, unless, that is, you're bound to a nuclear import receptor. What the nuclear import receptors can do is break the weak hydrophobic contacts that maintain this hydrogel on the interior of the nuclear pore complex, and rapidly traverse the nuclear pore complex and arrive in the nucleus. And so this ability to break these weak hydrophobic contacts is also something that the protein disaggregates has to do when it's pulling proteins out of an aggregate, and made us wonder whether cat beta 2 might also have disaggregase activity. I should note here that the interaction between cat beta 2 and FUS is regulated by a small GTPase called RAN. In the cytoplasm, we find RAN in the GDP bound state, which permits this interaction. But as soon as this complex arrives back in the nucleus, it encounters RAN in the GTP bound state. And RAN GTP engages cat beta 2 and causes cat beta 2 to eject FUS. So now FUS can go and perform its regular function inside the nucleus. And so initially we wanted to do a very simple experiment. And what we're able to do is we're able to assemble full-length FUS into these amyloid-like fibrils in the test tube. Now these are fairly stable structures. So if we simply dilute these FUS fibrils with buffer, they're stable for many hours, days, and weeks in the test tube, in fact, and don't spontaneously dissolve, suggesting that they are rather stable structures. Moreover, we think these structures are bad news. So if we drop these structures onto neurons in culture, in collaboration with Don Cleveland's lab, we've shown that these are highly neurotoxic structures. Moreover, in collaboration with Don Cleveland's lab and Sandrine de Cruz's lab, if we inject these fibrils into the brains of mice, the mice begin to develop an FTD-like phenotype that is caused by the prion-like spread of these fibrils and the conversion of endogenous FUS into these fibrillar structures. So they really have prion-like activity in that sense as well. So what we're going to do here is we're simply going to add cat beta 2 at equimolar concentrations with these FUS monomers in these fibrils, and come back a few minutes later and look to see what's happened. And so remarkably, within 10 to 20 minutes, cat beta 2 rapidly disperses and solubilizes these FUS fibrils. And actually, we don't have anything else that dissolves these FUS fibrils quite as rapidly as cat beta 2. This activity depended on the interaction between cat beta 2 and FUS. So if we mutate two tryptophans to alanine, that weakens the interaction between cat beta 2 and the FUS PY-NLS, then we're no longer able to disaggregate these FUS fibrils. Moreover, if we take a parallel nuclear import receptor system importing alpha and cat beta 1 that doesn't recognize the FUS PY-NLS but recognizes the NLS in TDP43, this nuclear import receptor system is unable to dissolve FUS fibrils, suggesting a very specific activity of cat beta 2. This activity is regulated by the small GTPase RAN. So if RAN is in the GDP bound state, this disaggregation reaction is permitted. But if RAN is in the GTP bound state, then disaggregation of FUS is inhibited, suggesting that this activity is likely to be restricted to the cytoplasm of cells. And so this activity was very surprising to us. So we wanted to do a few more controls to really assess whether this was a specific activity. So here are two further controls, two proteins HDAC1 and a FUS antibody raised against the PY-NLS of FUS that both engage FUS with high affinity. But neither of these proteins can dissolve these preformed FUS fibrils, suggesting something very special about the interaction between cat beta 2 and FUS. And so what do we think is going on here? So FUS fibrils are maintained by intermolecular contacts between prion-like domains that hold the fibril together. And this presents the PY-NLS of FUS on the surface of the fibril, which is then engaged by cat beta 2. But this binding event alone is not sufficient to drive disaggregation of the fibril, because otherwise the FUS antibody that also recognizes the FUS PY-NLS would also dissociate fibril. So it's here that we think that Cat beta 2 makes secondary contacts with FUS that breaks the intermolecular contacts that hold the FUS fibrils together. And it's this activity that we believe is related to the ability of Cat beta 2 to traverse the nuclear pore complex, where it breaks weak hydrophobic contacts. But in this context, enables FUS to solubilize these FUS fibrils, and ultimately we're left with heterodimers of Cat beta 2 and FUS, which we believe are competent for nuclear import. So how do we know that's the case? So if we take those soluble disaggregated products and pull down either Cat beta 2 or FUS, we pull down all of the Cat beta 2 or all of the FUS. Unless we add RAN GTP to those soluble disaggregated products, what RAN GTP does is it dissociates FUS from Cat beta 2. And we believe that then in the context of the cell, Cat beta 2 can dissociate FUS in the cytoplasm, transport it into the nucleus, where RAN GTP completes the disaggregation reaction by dissociating Cat beta 2 from FUS. FUS can now go perform its regular function inside the nucleus. And so this activity also extends to these hydrogel structures that we can make of FUS, which are formed by dense networks of FUS fibrils. Cat beta 2 will dissolve these hydrogel structures within an hour or two, whereas our negative control Cat beta 2 with these two tryptophans mutated to alanine will not be able to drive this process. So Cat beta 2 can really reverse the solid phases of FUS. But then what about the liquid phases? So under different conditions, we can assemble FUS into liquid droplets in the test tube, and we know that these are really liquids. Here are 4 droplets fusing over here, indicating liquid-like properties. And so we're going to add in Cat beta 2 at equimolar concentrations with FUS. And so we just added it right there. And what you'll see is that these FUS liquid droplets are dissociated within a few seconds and minutes of adding Cat beta 2. If we do the same thing with a mutant form of Cat beta 2 that doesn't bind the FUS PYNLS as tightly, then we're unable to dissociate these preformed FUS liquid droplets. So that's really exciting. This also works for FUS liquid droplets formed by disease-linked mutant forms of FUS. And so in collaboration with Sue Myong's lab at Hopkins, what we've done here is form these liquid forms of FUS, which form liquid droplets much more aggressively than the wild type. But once they form, we add Cat beta 2 and we rapidly disperse these structures. So that's exciting. That's all at the pure protein level. What about in cells? And so together with Aaron Gittler's lab, we developed a yeast model of FUS proteinopathy, where overexpression of FUS causes FUS to aggregate in the cytoplasm of yeast and is also very toxic to yeast cells, phenocopying what we see in degenerating neurons of ALS and FTD patients. And in this experiment, what we're going to do is we're going to allow those aggregates to form with Cat beta 2 switched off, but then we'll switch off FUS and switch on Cat beta 2, so we can be confident then that any solubilized material will have come from aggregated FUS and not newly synthesized FUS. And what we see in that case then is that Cat beta 2 obliterates these cytoplasmic aggregated forms of FUS and restores FUS back to the nucleus. And this is a phenotype we'd really like to achieve in ALS and FTD patients because we believe this would be highly therapeutic. This activity was not driven by this mutant form of Cat beta 2 that has a weaker interaction with FUS. And so this is in yeast. What about in ALS patient-derived fibroblasts that bear this mutation R521H in the FUS gene? Here what we noticed is that already in these ALS patient-derived fibroblasts, we're seeing some FUS mislocalization to the cytoplasm and a reduction in the expression level of these 3 FUS target genes. And so if we introduce Cat beta 2 though into these patient-derived fibroblasts, we restore FUS back to the nucleus and rescue the expression of these 3 FUS target genes back to levels that we see in control fibroblasts, really suggesting that we are rescuing the loss of nuclear function of FUS through this activity of Cat beta 2. Moreover, this activity is neuroprotective in flies. So in collaboration with Uday Pandey's lab at the University of Pittsburgh, what Uday has done is generate a fly model where he expresses this mutant form of FUS only in the motor neuron of fly. This leads to a very severe motor neuron degeneration and a reduction in fly lifespan. If we co-express an innocuous protein like GFP, this has no effect on this phenotype. But if we express cat beta 2, then we are able to significantly extend fly lifespan. It's not a complete rescue. We're probably not hitting quite high enough cat beta 2 expression levels, but if we could have a therapeutic that worked this effectively for ALS and FTD, this would be something that we currently don't have. So finally, what about TDP-43? So TDP-43 is recognized by a different nuclear import receptor system. So important alpha engages the TDP-43 NLS. Important alpha in turn is recognized by cat beta 1, which transports TDP-43 into the nucleus. If we preform TDP-43 fibrils in the test tube, then cat beta 2 is unable to dissociate TDP-43, and here serves as a negative control because it doesn't recognize the specific NLS sequence of TDP-43. But adding important alpha and cat beta 1 is able to slowly dissociate these TDP-43 fibrils in the test tube and works about as well as one of our engineered HSP104 variants in vitro. Then in collaboration with Wilfried Rossel and Diego Lincoln Rimas at the Mayo Clinic and at the University of Florida, what Diego's lab has been able to do is to assess whether the fly homolog of cat beta 1 is also neuroprotective against TDP-43 proteinopathy in the context of the fly. So overexpression of the fly homolog of cat beta 1 suppresses TDP-43 toxicity in the context of the fly eye, whereas RNAi enhances that toxicity. If we express an ALS-linked mutant form of TDP-43 in the fly brain, we begin to see TDP-43 inclusions, but this is mitigated by the expression of cat beta 1 in fly, and that translates into a locomotion activity as well, suggesting that upregulation of nuclear import receptors are also neuroprotective against TDP-43 proteinopathy. So I'll conclude there. And so what we think is happening in disease is that FUS, for example, initially experiences some form of nuclear import defect that causes it to accumulate in the cytoplasm. Then upon some stress, FUS will perhaps condense into liquid phases, and these will ultimately convert into the fibrils that we associate with the end stages of disease. But what we can do with cat beta 2 is essentially reverse all of these steps. So we can disrupt those solid phases, these liquid phases, and restore FUS back to the nucleus. And so I've told you that cat beta 2 can potently prevent and reverse FUS aggregation, and the parallel system for TDP-43 can also have this same activity. Cat beta 2 disperses the FUS liquid drops and reverses the aberrant phase transition of FUS, so it can dissolve these preformed FUS hydrogels. And the final product of that disaggregation reaction in the test tube is a complex between cat beta 2 and FUS, which can be transported to the nucleus and dissociated by RAN GTP. And that elevating cat beta 2 expression mitigates motor neuron degeneration in the fly due to this ALS-linked mutant form of FUS. And so this very much changes our view of the nuclear import receptor. They're not just nuclear import receptors. They function as a potent chaperone and disaggregation system in the cytoplasm, specifically for their cargo. And that nuclear localization sequences are not just nuclear localization sequences. So in the context of the cytoplasm, they're functioning as anti-aggregation or disaggregation signals by recruiting nuclear import receptors to them to bind. And here it's worth noting that about 3/4 of proteins that have a prion-like domain in the human genome also have an NLS sequence, suggesting that this is a broad phenomenon used by these RNA binding proteins with prion-like domains to mitigate their aggregation in the cytoplasm. And finally, this suggests that AAVs or other methods, such as small molecule drugs or small activating RNAs that enhance cat beta 2 expression, or other nuclear import receptors could be valuable therapeutics for ALS and FTD, where typically we see that nuclear import receptors are expressed at lower levels. And so I'll stop there. I'm grateful to various funding sources and my lab, and we have a number of great collaborators that I mentioned as I went through the talk. I particularly want to shout out Ling Guo, who really led the cat beta 2 work, and now has her own lab at Thomas Jefferson. So I'll stop there, and thanks for listening, and I'll take any questions.
Thank you very much for the great talk. Let's see if there are any questions coming. Okay, so while people are typing, I have a question for you in regard to the binding of the disaggregates. You show that it binds on the side of the fibrils first, of TDP-43 fibrils, and then later on binds on where is the core of the fibrils. Do you think that there are two binding domains? Any speculations more of a molecular mechanism?
Yeah, so work from our collaborator, Yumin Chuk and Mike Rosen have used NMR to start to map those secondary contacts with FUS and cat beta 2, and that it seems like multiple heat repeats within cat beta 2 are making these secondary contacts with other domains of FUS. Yeah, so we started to map those, but we don't have quite the same level of understanding of those quite yet.
And the other thing which I'm kind of trying to wrap my mind around this, I have never seen, maybe I missed it, but this is quite remarkable in disaggregating the fibrils. But you know that the fibrils are really stable because of all these hydrogen bondings, and there's a huge energy penalty for disaggregating it. Any thoughts on, again, it's more from molecular side?
Yeah, no, it's a great question and something we're working to understand, right? But in essence, what the cat beta 2 must be doing is making it more favorable for the FUS to be soluble than in the fibril state, right? And so what's becoming very interesting is that this is an ATP independent process, right? This is all through binding interactions, but cat beta 2 is joining a growing class of ATP independent chaperones and disaggregases, which now include some of the prolyl isomerases like cyclophilin 40 that Laura Blair and Chad Dickey have shown can dissociate tau fibrils, but also the trim proteins that our collaborator Xiaolu Yang has shown can dissociate alpha-synuclein fibrils, and more recently these proteins that contain a polyacidic stretch such as DAX, which also have some ability to dissociate these fibrils. And so the energetics of these processes are very interesting to think about, because it initially seems very unfavorable, right, to convert the fibril to the soluble form, but that's what these agents are doing, and figuring out exactly how that's working is going to be fascinating, I think.
Yeah, thank you so much. That's a really fascinating. So we have questions already coming. The first one is from Christian Grissinger. Thanks for the talk. I could imagine that the nuclear transport of receptors are not specific just for FUS or TDP-43. By having more of these to disaggregate FUS or TDP-43, is there not the risk or disbalance of proteins between cytosol and nucleus?
Yeah, that's a good point and something to think about. So by upregulating their expression, we would shift proteins more towards the nucleus and away from the cytoplasm, where perhaps there is some optimal balance for that that nucleus cytoplasmic ratio. The thing that's making us sort of excited about developing these as potential therapeutics is when we deliver them in AAVs to a mouse model of FUS proteinopathy, for example. We're not having damaging off-target effects, and so that doesn't seem to be, you know, a major problem, at least in these FUS transgenic mice, for example. But it's certainly something to think about, and perhaps we wouldn't want to express them at high levels long term. We just want to express them long enough that we can reverse the pathology and then get back to normal.
So the next two questions are from anonymous attendees. The first one, hi, very nice talk. I'm curious that as a therapeutic point of view, does development of kappa beta activator restore FTD symptoms? Another one is, does the aggregation of TDP-43 has some connections with stress granules?
Yeah, so the first point, yeah, we can antagonize some of these FTD-like phenotypes. And then the second question is TDP-43 connected to stress granules. So yeah, great question. I think there are multiple routes to TDP-43 aggregation in the cytoplasm. One of those possible routes is through the stress granule. And I think Paul Taylor's group showed that very nicely in that optogenetic induction of stress granules through a Cry2 tagged G3BB1 protein. Those stress granules will ultimately convert to a TDP-43 inclusion. But it's not the only way. And so TDP-43 can aggregate independently of stress granules in the cytoplasm as well. And which mechanism is predominant in disease is something that's not clear at the moment.
The next question, there are a couple questions, the second anonymous attendee. What is the concentration dependence of kappa B2 effect on amyloid aggregation? I think may have been spelled. And the second one, did you check kappa beta 2 with other amyloid proteins such as tau and alpha-synuclein?
Yeah, it's really a stoichiometric disaggregate. So one kappa beta 2 pulls out one FUS under the conditions we've reconstituted it at. And so it begins to max out as you approach stoichiometric concentrations. And then what we haven't done though is to try and make the system catalytic by including RAN GTP in there. So you recycle kappa beta 2 for further rounds of disaggregation. And in that way, we think the system as a whole inside the cell can be pseudocatalytic. Then other amyloid proteins. So unless you have an NLS sequence, these proteins will ignore you. So they can dissociate other cargoes that assemble into amyloid fibrils that have an NLS. So proteins like TAF15, EWSL1, H&RPA1 and A2. But tau and alpha-synuclein don't have the same type of NLS and so are not affected by these proteins. But one thing we're trying to do is put an NLS sequence on synuclein and tau and then see if this machinery can recognize them.
Thank you. So the next question is from Aiton Lerner. Great talk. Could it be that what drives the disaggregation within the gel-like condensate is the formation of two-component condensates, FEUs and the disaggregate, to allow their effective interaction and shift back to solid state?
Yeah, that's an interesting question.
There's a couple more things after that, if you.
Okay. Yeah, we haven't thought too much about that. I mean, our current thinking is that kappa beta 2 pulls FUS out of these phases one by one, right? And so yeah, it's interesting to think about how that might affect the phase as that is happening.
Yeah. So there's another question coming, and I'm reading it from the chat area from Victor. Hi, Dr. Shorter. I'm not very familiar with FUS, so just curious, what's preventing it from aggregation within the nucleus?
Yeah, great question. So inside the nucleus, what we think is happening that FUS engages its RNA clients, its RNA partners, and this is really very effective at stopping FUS aggregation inside the nucleus.
All right. Thank you very much. I think it's about time to open for the general discussion. And please, the audience, raise your hands if you want to ask your question.
I have a question for
Eri.
James.
Okay, go ahead, Danilo, and after that we'll go to Eri's question. Yeah.
Sorry. No, please.
No, please go ahead, and after that we'll.
Sorry for that. So my question is this: this phase transition, droplet gel versus fibrillar order aggregate somehow resembles an order-disorder physical transition. So I wonder whether if it would be possible, at least in principle, to rationalize this transition in terms of physical properties like entropies or entropy gain, and relate the effectiveness of inhibitors within this framework, in your opinion?
Yeah, absolutely. That would be neat to frame things in those terms. Yeah.
Okay. Thank you.
I guess I'm going to read the question. It's from Anup Arunagiri. This one question is for Eri. Nice talk, Dr. Chatani. You mentioned briefly in your talk that the environment could influence insulin aggregation. Have you tested any physiologically or pharmaceutically relevant conditions that may influence B chain self-assembly?
Thank you for your question. This time, we showed the result at only alkaline pH, but we also tested other pH, including neutral pH. We expected some influences by pH, but the fundamental property was very similar among all pH. But we could see protofiber formation proceeds differently at weakly acidic pH. So probably depending on it means the fibrillation nucleation pathway is influenced by the outer environment.
Thank you so much. I promoted Carmelo to the panelists. Carmelo, if you want, you can ask your question.
Yes, thanks, Magda. I have a question for James. I'm curious to know if you have investigated if in the protein in your droplets is in equilibrium with the free protein outside the droplet.
Yeah, of course, there's an equilibrium there, right?
Okay, thanks.
Thank you so much. Rams, Sam, do you have any questions?
No, great talks. I have questions, but I think we are running out of time. So maybe I can ask one short question to Dr. Chatani. How did he come up with this idea of ultrasonic pulse? Does this work for other amyloids too? Or what happens if we continuously apply this pulse in the aggregation process? Or when do you apply? It seems to be very specific in terms of time and so on. Yeah.
Thank you for your question. So I began to use the ultrasonic pulse when I was staying at Professor Goto's laboratory. So he often used ultrasonication as a kind of agitator, and he found it enhanced fibrillation very, very effectively. So probably the main reason for such acceleration is probably due to that ultrasonication can make air-liquid interface by making bubbles, and its collision and its disappearance disturbs air-liquid interfaces, and thereby protein structure will be deformed, stimulated. So that's why aggregation is accelerated by ultrasonication.
Thank you. Thank you.